Hello, hello everyone. Welcome you all to my channel Igniting Curiosity in Life Science. So this is Savita. So today we will explore another very interesting and important thing in all the eukaryotic cell, that is the nucleus. Okay. So what is nucleus? Nucleus, it is situated in a deep inside every eukaryotic cell, and it lies a remarkable command center. Okay, that is what a nucleus, and it is called as a true nuclear true control of a hub of all the life activities. So what it mainly contains? It contains most of the genes. Okay, so, in almost all eukaryotic cells, some genes are also present in the mitochondria and the chloroplast. The nucleus, it also stores most of the genetic information. Okay, that is what the DNA. It is the master library that holds the instructions to build and operate the entire cell. Okay, and the nucleus, it is having so many the parts. So nuclear pore, okay, nuclear envelope, nuclear lamina, nucleolus, okay, chromatin and the nucleoplasm. Let's see one by one what are the main functions of those, and what is the main structure of the thing. Okay, then coming to the nuclear envelope. So nuclear envelope is a double membrane structure, okay, and it protects the nucleus. So the space between the two layers, it is approximately 20 to 40 nanometers in wide or width. Okay, the envelope is dotted with the nuclear pores of about 100 nanometers in the diameter. So these pores mainly act like a guarded gateways. Okay, it is allowing only certain proteins and RNA molecules to pass in and out and maintaining order inside the nucleus. So the nuclear pore complex, here you can see that the nuclear pore complex, okay, it is having several copies of a pore proteins, okay, of about 30 in number, which is also called as what the nucleoporins or NUPs. Okay, so the transfer of a molecules by nuclear pore complex is mainly carried out by two mechanisms. Okay, what are the mechanisms? Here you can see that the passive diffusion as well as the selective transport. So what is passive diffusion? Some molecules, especially the small molecules with less molecular mass of up to 40 kilodaltons, they move freely across the pore complex by the diffusion process. Okay, and in most of the macromolecules like RNA and proteins, they use what the selective transport systems for their movement across the pore complex. So generally what happens, selective transport is triggered in response to the specific signals which regulate the transport. So nuclear pore complex, it controls mainly the movement of specific molecules across the nuclear envelope, and it regulates the protein transfer essential for the gene expression, as well as such as for example the transcription factors, okay, synthesized in the cytoplasm. So this is what the electron micrographic the picture of a 3D picture. It shows what the main structure of a nuclear pore. Okay, so it is having the main structure. It is showing the nuclear 8 spokes connected to the cytoplasmic rings. Okay, here what the cytoplasmic rings. So it is mainly connected to the 8 spokes and anchored at the junction of the inner and the outer the nuclear membrane. The NPCs or the nuclear pore complexes, it forms what a basket like shape. Okay, here you can see that the basket like shape. So it is mainly channeled by the FGNUP proteins. So what are these? So FGNUP proteins are a special group of proteins, okay, found in the central channel of the nuclear pore complex. Okay, they are mainly rich in the amino acids phenylalanine as well as the glycine. So phenylalanine here, it is represented as the letter F, and glycine, it is represented as the G. So together it forms what a FGNUPs proteins. Okay, these proteins are mainly rich in the glycine and the phenylalanine, which creates a selective barrier controlling the nuclear transport. So what is the main function of this? That is what a guarded gateways. Okay, the main function of this, it mainly transport. Okay, during transportation, it selects the selective, it acts as a selective permeability barrier. Okay, allowing only the specific molecules to pass between the nucleus and the cytoplasm. So smaller molecules, they diffuse very freely, but larger molecules, it need what a nuclear transport receptors, okay, like importins and the exportins to pass through. Okay, so it also acts as the second point, it also acts as the gatekeeping role. That is, they interact with the transport receptors, okay, that recognize nuclear localization signals. Simply we can call it as what the NLS. Okay, so what this will do, the NLS. So NLS, it also called as what the nuclear export signals or nuclear localization signals on the proteins. Okay, and as well as this ensures that only authorized macromolecules enter or exit the nucleus. Okay, this is the second function. And third function, it maintaining the NPC integrity. So what is that? It is mainly helpful for the stabilization of the nuclear pore complex structure and maintain its dynamic nature for the continuous transport of the molecules, okay, in and out of the nucleus. So next we'll move on to the nuclear lamina. So nuclear lamina and the matrix. Nuclear lamina, it is the protein network supporting the envelope. Okay, that is what it is, lies just beneath the envelope, lies the nuclear lamina, a supportive protein network that gives what the nucleus its main shape. Okay, inside a nuclear matrix helps organize the DNA so it can function very efficiently. Okay, within the nucleus, DNA combines with proteins to form a chromatin. Okay, this is what a chromatin, here you can see that this is a chromatin. How it is formed? DNA plus the protein. Which protein? That is the histone protein. Okay, it forms what the structure, the chromatin. So again when a cell prepares to divide, this chromatin coils very tightly and again it forms what the chromosomes, right? So each species, okay, after that each species it forms. So this is what a simple structure of a chromatin and the chromosome. Okay, this is a double helical structure of a DNA. Here you can see that. So double helical structure of a DNA and beads on a string of DNA wound on a nucleosomes. That is what it forms with the histone proteins. It forms what a DNA octamer. Okay, then chromatin fiber, then very condensed form of the nucleosome. It forms what the chromosome. Okay, DNA plus protein, chromatin, and condensed form of a chromatin. It is what a chromosome. Okay. Example, inside the nucleus means every species has its own unique chromosome number, isn't it? For example, in the case of humans, Okay, he has the 46 chromosomes in the diploid or the somatic cells, whereas in case of the sex cells or germ cells, it is having the n number of chromosomes, that is the 23 chromosomes. Okay, in case of the fruit fly, 8 in the body cells or the somatic cells, whereas in case of a haploid, that is what the in the germ cells, they are having the 4. So here 46 chromosomes in case of a diploid, 2n. Okay, whereas in case of the n, that is 23 chromosomes. In case of a fruit fly, 2n. Okay, 2n it is 8 and n it is 4 cells. Okay, and next coming to the nucleolus. So what is nucleolus? Nucleolus, it is also a main site for the RNA synthesis, okay, and the ribosome assembly. So DNA, it gives what the instruction to make or to prepare what the rRNA, that is what the ribosomal RNA, it again forms what the ribosomes. So rRNA plus proteins from the cytoplasm, it forms again what the ribosomal different subunits. 2 subunits are there, ribosomal, and subunits exit nucleus, okay, combine into ribosomes in the cytoplasm. So this is what a picture, it's showing what a from DNA to protein, how it is going to be formed. So in this case, you can see that the DNA, okay, it is mainly occurred in that transcription process. Okay, DNA, it is going to form mRNA, and in the translation, it is going to prepare the protein. Okay, DNA to mRNA and mRNA to ribosome and the protein. This whole we can call it as what this process, the central dogma. Clear? This is the thing. And nucleus, mainly it makes the mRNA, that process we can call it as what the transcription. Okay, and again from the ribosome to the protein synthesis. Okay, that process we can call it as what the translation. So at last we will discuss what the main functions of the nucleus. So what are the main functions? First thing, it stores the genetic information. So it contains mainly the DNA, okay, which holds all the instructions or the characters for the cell's structure and the function. And also it acts as the blueprint for building proteins and also controlling the cell activities. As well as the second point is it controls the cellular activities. How? Mainly through the regulation of regulation processes like growth, metabolism, okay, protein synthesis, as well as the cell division. And also it ensures every function in the cell, okay, happens in an organized and timely way. Okay, next point is the regulates. It regulates what a gene expression. So it mainly determines which gene are turned on and turned off condition. It is mainly deciding what proteins are made and when it should be made. Okay, and the second point is this mainly regulates, helps cells perform specialized functions like muscle cells, okay, nerve cells, or the skin cells, like that, or brain cells, okay, like that. And next the main function is it directs protein synthesis. So DNA in the nucleus is transcribed into mRNA, that is what the messenger RNA. So mRNA exits through nuclear pores to ribosomes, okay, where it is translated into proteins, that is what the translation process. So next function is the ribosome formation via nucleolus. So the nucleolus, inside the nucleus, it makes what the ribosomal RNA or rRNA and assembles ribosome subunits. Okay, these subunits then move to the cytoplasm for the protein production. Okay. Next, the function is it maintains what the genetic integrity. So mainly the nucleus, it protects DNA from damaging using the nuclear envelope, okay, and also it repairs what the enzymes, okay, and also it ensures accurate DNA replication, okay, during what? The cell division. Yeah. And the function is it coordinates the cell division. So during mitosis and meiosis, the nucleus, it ensures that the genetic material is evenly distributed to the two daughter cells, isn't it? So that is mainly contributed by what? The nucleus, okay. And nucleus, it is we can call it as what? The heart of a cellular control. Okay, where life's blueprint is stored and read, and it also it is safeguarding the genetic instructions and guiding the creation of life's building blocks. What are those? Yes, the proteins. Okay, this is what all about the today's content, that is what the eukaryotic cell nucleus. Okay, so that much about the today's class. So this concept is also helpful for the KSET or CSR NET and the NET exam, UTC NET exams also. So keep focusing and keep preparing for the other this competitive examination. So stay curious and keep exploring the world within us. Okay, only in the channel Igniting Curiosity in Life Science. So keep watching. Thank you. Please like, share and subscribe this channel for more the life science the videos. Thank you.